Hello and welcome everybody to our next YPIC webinar with Maarten Dhaenens from Ghent University. Maarten is our former YPIC president, so we are super happy that he's here and gives this talk. Scientifically, Maarten is very interested in the histone code. He's also the team leader for proteomics and mass spectrometry at the University of Ghent and the coordinator of the proteomics service facility. He stopped his interest in the histone code for a few months to develop an analytical platform for Corona in collaboration with industry, and that is what he's going to talk about today. So with that, we are super happy that you're here, Maarten, and the stage is yours. Please share your screen.
Yeah, thank you, Florian, for this nice introduction. Yeah, so this is a YPIC presentation, so it's a bit different from the normal scientific stuff, I guess. But as the title already says, a blind date with industry in Corona Tavern, just a fling or the start of a romance. I'm not going to explain you how to come in contact with industry and how to make sure that you have a nice romance. I'm just going to tell you my story, and I hope that it helps you in growing and drawing your own conclusions. So I'm actually in no means an expert in this, but let's get to it. I mean, I've been working on developing the lab based on 3 different pillars. That's easy to get your head around if you know what things are you need. In my case, that was looking for funding. That's what everybody does. You need scholarships to be granted in order to pay people and buy instruments. We also founded a service lab called Progentomics, where we help other academic labs with proteomics services. And that 3rd pillar always has been industry. As you can imagine, it's something very appealing. They always have the newest stuff. They're always ahead of where we are, and teaming up with them is just a very tempting thing to do. And I'm talking from discounts all the way to true hardware beta testing. They can help you in many ways. Now, irrespectively of which pillar you are focusing on, you'll always need to be an expert in something to get your grants. They want to see you as the expert, and it helps if you have a niche. So it's not necessary, but it helps because then the competition is less. And I think collaboration will always be a very important part in attaining anything in science. But so how can we try and convince industry to join us on a date? Well, I know they want things to be applicable, say some clinical things, forensics, food, environment, these things that have a large market. They want things that are and user friendly. So nobody really is into developing anything. That would be our task in this case. Well, preferably it would be new and trending, obviously. Then it's easier to sell in the end. And if you're doing anything fundamental, make sure that it's embedded in the conventional way of doing things, so that the threshold is not too large. Well, this is, I guess, the theory. Now, in a relationship, there's 2, right? So there's the industry, and in this case, I'm saying me. I have my own type, so it's going to be about mass specs, as you might have imagined. And I have a very specific type to date, and that is a QTOF. I can't help myself. I just love QTOFs. And so I guess that limits a few companies that I would never be able to date because they're not my type. And that doesn't say anything about their qualities and whatnot. It's just I'm narrowing it down already. Now, just before this quadrupole became a prize for YPIC, and this TOF cube became the most expensive plant pot in Europe, they generated a spectrum now about 11 years ago. And it was Pauline Meert, a student of mine, who did it. And this is the deconvoluted spectrum, as you can see it, as it was generated back then. We're around 15 kD. And it's like a Gaussian mountain of peaks, 14 Daltons apart. Now, that kind of was the point where I realized, okay, if it's Gaussian, it must be very complex. If not, it's not Gaussian. And on the other hand, everything that's random is Gaussian. So is this a random thing? So it's complex and random, but it shouldn't be random because it's histones, as you might have guessed by now. This is H3. And I felt like this could be that one thing I could build my career on. And who knows, one day bring these flowers to industry and become an expert. Bam, histones. They are, according to us at least, very cool molecules. Very recently in Quantum magazine, on the upper right here, there was an article on histones with the illustration, this one from Jason Lyon. I like it a lot. It's a bit explosive, to say the least, but what it shows you is that they lay at the foundation of basically any animal or plant we can visibly see. The eukaryotes, that is. So, briefly walking you through the basics of histones. There's a recent theory saying that it were the archaea that contributed histones when they melted with bacteria. And together, eukaryotes arose. And this is about 2 billion years ago, I guess. 2.5, some say. And so in this way, all eukaryotes we know today came into being. Now, what these histones did is they helped manage the information in the DNA. It's such a long stretch, that DNA, that it needs to be regulated and packed. Now, the way histones do that is they have a histone code, which is basically a language of modifications that change the interaction between the nucleosome, so that's the structure the histones form, and the DNA, so based on charges and many other processes. And in this way, if anything opens up, a protein can be transcribed. And in this way, histones help regulate what proteins can be expressed in your body and especially where they are being expressed. Now, what I only recently found out is that you remember how bacteria were contributing the energy and the archaea were providing a mechanism to cope with all the information. Basically now, many people, and I kind of agree, think that histone post-translational modifications are in fact the metabolic sensors. So they translate the metabolism into the DNA, into, well, the activity of your genes. For example, if a cell is happily metabolizing with the Krebs cycle, then it's probably going to put PTMs on histones that open up the DNA so that a lot of transcription can occur. But as soon as other metabolic pathways need to be used in order for the cell to survive, they start to shut down transcription to save energy. And so it's a dynamic mechanism that stems 2 million years back, wherein metabolism is being translated in the information. And you can imagine, if you know how many molecules are in these huge biochemistry charts, that it's a pretty complex language. It probably is, we don't know yet. But I even feel like there might be a grammar to this language. And so while DNA is something that is being sequenced, and I don't want to be disrespectful, but it's being read out. So there's the letters coming out of the instrument, and then all the bioinformatics is done on the letters. This is not something we can do with histone PTMs or proteomics for that matter. We cannot read it out. We have to use LC-MS systems, which, as you've guessed, in our case, QTOFs, but these generate numbers. They don't read out anything. They spit out numbers. And we need to add another level to the data analysis before we can start thinking about biology. We need to interpret these spectra before anything else. So we have an extra kind of burden or step, if you like. But now the thing is, mass spec has been used for proteomics for a long time. And so that's pretty well fine-tuned, and it works very well if you're doing what we call proteomics. But for histones, we are actually looking at only 5 proteins and their variants. And we're not looking at the amino acids, we're trying to decipher the alphabet that sits on top of that. So it's an additional layer. And yeah, that kind of makes it a lot more complex, because not many people know this, but at least I'm saying over 26, because the normal alphabet has 26 letters. But I think by now, probably 30 different groups have been found sitting on histones. So even the most modest estimations say that these 5 proteins, with all their possible combinations, can yield easily 70 million different RHC-like peptides. So we don't have much to do with conventional proteomics anymore. We just are left with a totally different set of challenges, which we need to try and tackle one by one. And this is the only technical slide here I'm sneaking in. So the problem is histones have a lot of lysines and arginines. So we need to chemically derivatize them before we add trypsine. Yeah, there's basically so many PTMs that there is no supercomputer on Earth that could combine them all in any search space. So we have no way to search all the PTMs. Well, most scoring algorithms at least have some intensity aspect in them. And so more often than not, the actual fragments that can distinguish 2 differently modified forms are too low to be picked up by the scoring, and we have tons and tons of ambiguity. Now, to make matters worse, a lot of these combinations of PTMs are obviously isobaric, others are near isobaric. They often co-elute, and so they get co-selected. So we get a lot of chimeracy. And basically, to date, we don't have a decoy strategy. So we don't even know how well we're doing in terms of false discovery rate. And finally, I think one more important aspect is if we want to be quantifying specific PTMs, we need to try and extract the amount of that PTM from a set of different peptide forms or peptidoforms, and that is not an easy thing to do. So what we do know is we have a niche, and we aim to become an expert in it. It's called histones. But looking at the nice bouquet of flowers that industry would be tempted by, it turns out that we didn't make a very good choice at all, actually. Applicable histone studies, yeah, it will be in 10 years. I'm sure, I'm convinced of this, but not quite now. We're just still figuring out how to measure them. So is it end user friendly? Well, I've only seen terrified end users so far. Most just run away and ask us to do it because it's just too frightening. New and trending, 2 billion years is not really new and trending. Fundamental, that it is. But the problem is we study an entire different alphabet altogether. So it's not embedded in conventional proteomics. And if you ask anybody about histones who never did it before, they won't know where to start. So it's not a big market. It's just not the right bouquet we're holding here. But it's not all about pleasing industry. On the contrary, actually, this is not my point at all. I'm just going to tell you that you just have something in mind, you want to study these histones, then do it. And that's what we've done. We kind of started all over again because we do want to become experts at least at one point in our career. So we started from scratch. We started extracting histones, but then specifically in order to minimize technical variation for label-free quantification. We then started accidentally detecting this chemical noise I was mentioning from the derivatization process. And we started looking into how we could solve these problems and reverse certain certain chemical artifacts. Very recently, we basically even tried to provide people that are new to the field a roadmap to sample preparation, because there's so many ways you can go about this, and we have this kind of easy tool to or a tool to get an easy overview of what PTMs you're actually seeing at any given time. We basically try to curate the search. Like I mentioned, the combinatorial explosion is just so humongous that at one point we tried to suggest how you could be wise about your choices and how to curate PDMs. And during all that time, there was something I was really fascinated about, was trying to incorporate ion mobility and HDMSE into all this work, which was a collaboration with Waters. And it was actually the first time that we managed to get something on paper. I would say we were just very happy and lucky that Waters was happy to support us in trying to implement ion mobility and HDMSE, and we got this kind of software beta testing statute where we're very happy with. But you'll see how I'm building up to how this is not a one day thing, because then we started elucidating biology with collaborators. That is because we had been so preoccupied with developing techniques. But soon it turned out that this works pretty well. So we started looking at mouse stem cell biology, and then more closer to house, we did quite an extensive screening of human embryonic stem cells. And we noticed that there was a very conserved signature between these two organisms. So basically, we're now thinking along the lines of a stemness histone fingerprint that might have been conserved, who knows, throughout eukaryotes, but that is something I'll be looking at in the future. And there is this other thing, since we're part of the pharmacy department, is the first steps towards an applicable, an application basically for all the histone work, is a toxicoepigenetics assay. So basically, it's a developmental toxicoepigenetics assay, because we work on stem cells, but we basically incubate stem cells with different compounds and see how their histones react. And that sounds like something that might be picked up in the future. Now, I'm adding the little Sykes ion comb here because all the data I'm talking about here was done on a Sykes instrument. But then above all, we realized histones would only, well, really work if we applied data-independent acquisition. And in 2013, I would say arguably a year where mainly Orbitrops were being sold, and many years before and after, we kind of chose to buy a Synap G2SI and a triple TOF. So you can see how two vendors already kind of popped out. And we started looking into DIA because I think this one image sums it up. This is an image of two peptides from histone H3, one acetylated at the K18, the other one acetylated at K23. And so you can imagine, if just this one group is switching, that there's ever so slight change in retention time or shift in retention time, but they weigh exactly the same. So on an MS1, you would just see one bump. But if you start looking into the transitions, being the fragments that help you to discriminate between these, you can just set them apart like nothing. So we were pretty much sure that DIA is the only way we're going to settle this in the end. So we heavily started to invest in DIA. This was basically the histone swath workflow, which took us years to really make work. Again, this is all the features and software they're meant to do proteomics. We need very different features in software and in many other places. So it's a very slow process. but we increased our efforts in trying to develop data independent acquisition. And I think one of the most exciting ones coming out of this was the AI networks. So we basically started to, well, preach, I would say, the fragment-centric revolution. To unify the proteomics field is maybe a very big thing. No, it's a very big thing. I'm not saying we're going to do it, but a man's got a dream. Basically, we're looking into trying to untangle co-eluding stuff in a new way by proposing a new data format, which is called AI networks. And that kind of uncouples your acquisition from your data analysis. So we're trying to develop a search strategy that can be applied to any acquisition strategy that's currently available. And we even want to propose a fragment-only acquisition technology, which is called SWIM-DIA on a waters instrument, pun intended. I've got this little Eve icon here to the lower left corner. This is just because it's so funny. Together with Leonard Maartens, we teamed up with a middleware company that actually does citizen science. And we're now working with this game called Eve Echoes. And they want to put an in-game challenge to the gamers to align these AI networks of ours. And so it must be, we don't know if this is going to work out, but if it does, then thousands of players will be aligning these AI networks, and we can get feedback from what they've done and try and understand the alignment process better. But the other thing that happened around the same time is the spectral predictions, now pretty commonplace, was something we wanted to incorporate in the DIA workflow for a long time. But we kind of managed that once we came across Brian Searle's chromatogram libraries and the narrow window DIA acquisition that is required to do so. And along with others, we managed to get predicted spectral libraries into the DIA workflow. And I think that was also a very nice one to have. And finally, I think one that is ongoing right now is, while we're trying to get the data across, is once you realize that there's so many different acquisitions and you want to figure out which one is most valuable for you, which software tools do actually work on this and do not and whatnot, was Bart, you can see on the right lower corner. He's a YPIC member, one of YPIC's friends, obviously, is acquired the same set of samples, which is the LFQ benchmark design from Navarro, with a QC on all these different kinds of instruments with all their different kinds of acquisition strategies. And so this is over 1,000 runs, which we're now trying to get across the internet to Proteome Exchange. And hopefully soon we can provide this bioinformatics benchmarks data set to everyone so that we can all better understand what's working where and how. So around the time that these plans were made, obviously, Corona struck. And this is where the whole Corona Tavern from the title comes in. Because at the time, there was only PCR, as everybody knows, to diagnose people. And especially the first few weeks, there was only little reagents to come by. So there was a lot of pressure on the testing facilities. Nobody was ready, nobody was prepared. And there were a lot of mass specs just sitting idle all over the place. And basically, if you come to think of it, a viral particle, it's built from, yes, RNA, but also from proteins and lipids. And especially two proteins, structural proteins, attracted our attention, which is the spike one, obviously the one piercing out, and then the NCAP nucleocapsid that actually wraps RNA. So that kind of reminds us of another molecule we know. We didn't realize at the time. but basically that increasingly is becoming interesting to be aware of. So there we went. That is to say, Bart again, same guy, now in a less fancy pose. He was just nagging on let's try this with the mass spec, and we have theoretical libraries to do non-model organisms, and we can do this. And I really didn't believe it at first, but hey, he was persistent and started doing this. And to our own surprise, when we did a very basic precipitation, a very short triptych digest, aiming for the clinic in the future at least, we did some swath runs and we applied the diamond diagram thing we just had published. Again, you can see we used the Sykes instrument for the discovery phase. That worked out just way better than we anticipated. So with the help of the Compomics group and all the predictions and so on, we came to quite a nice set of peptide markers pretty soon. And then we went 2 stairs up to another lab, and they had Exivo TQS. This stands for triple quadrupole. In our world, we were wondering, why would anybody put 3 quadrupoles in a single instrument? So just to say how far away we are from targeted. We were truly dedicated discovery people. We had no clue how to handle a triple quad instrument, so we had to reach out. And we tried this 8-minute sample, 8 minute per sample kind of flow. And when we saw the first results, and this in the x-axis, the CT values, and in the y-axis, the summed log intensities of all the peptides that we had selected in the discovery phase. Okay, no need to say, we were pretty much convinced that that this could be something. But yeah, there we were. We didn't know anything about PCR, and we didn't know anything about MRM. And if you come to think about it, these are 2 very different worlds. So this slide is trying to sum up what actually we were up against. Yeah, nasal swabs, they're nasty things. There can be a lot of other stuff in there, from blood to mucus, and they were being transported. They still are being transported in media of all kinds. You cannot imagine what people put in these media. Now, if you're doing PCR, there's a pretty conventional way of extracting RNA, and then you would amplify that in, well, basically an instrument that basically looks the same all over the world. And then they can get these CT values. I'm not saying this is a simple technique. I'm just saying it scales easily. And they end up with a detection of a CT value. So every CT, the RNA was doubled again. So the higher the CT value, the lower your initial viral load. But the frightening thing is that at a CT of 40, people are detecting 1 RNA molecule. So what about mass spec? There's a gazillion ways to extract proteins from anything. So we didn't know exactly where to start or what to optimize at first. There's all the proteins are in there. So it's a bit like looking for gold grains in a riverbed. You'll be shaking and you hope for the gold grains to remain on the sieve, but everything else is still in there. So that's how that works. We don't amplify anything. There is such a different, huge amount of different kind of instrumentations, and they all have their own software and LC systems and buffers and sources and you name it. So it's not like heating and cooling down anything. It's mass specs, right? So they're very heterogeneous, and we're not so much detecting as we are measuring in a way. So, I mean, most MRM results look like this, right? A lot of bumps, and you're just trying to figure out which one is the right bump. And in terms of sensitivity, well, I think we can reach 1 million molecules and probably quite a bit lower by now, but it's not 1 molecule at all. So the only thing that speaks for us is we have 300 NCAP molecules to wind up 1 RNA. So at least we've got that but it's not as straightforward as it might sound at first. And that's an obvious reason. Our world is more colorful, as I intentionally put here, but yeah, that means a lot of optimization. So when we first scaled up and started measuring patient after patient, it was very clear, and we had this one kind of workflow that we were using, and we were manually still picking out the peaks, and still, whatever we tried, there was a bend in the curve around CT20, and then it just flattened. So yeah, this is basically the point where we couldn't have moved any step further if it wasn't for the consortium we founded. Now, just to be clear, if you know that every CT value is a doubling, going from 20 to 40 implies a 1,048,576-fold increase in sensitivity. So yeah, we never actually aimed for 40, as you can imagine. But we were actually just in the discovery phase. It didn't feel that way, but that's, looking back at it, that's what it was. We had some recombinant proteins, which we had spiked in dilution series. We had a few patients. We had a few media we were looking at, and we had the discovery phase data and the first MRMs. And at that point, we decided to build a kit that we would then send out to as many labs as we could, so that everybody on their own instrumental platform could try and acquire the data and see how that worked in their hands. That, in the meantime, gave us the time to go back and see what we could improve in terms of sample preparation. And also in parallel, the Compomics group continued to do their magic. For example, machine learning on selecting the best peptide targets for diagnostic purposes and so on. And you can see how at this point in this consortium, all the vendors came back in our lives, and this time it was for real. Everybody had the same interest, right? So imagine this is about April, May last year. So there was nothing else to do but to develop a new assay. So everybody had their noses in the same direction, and everybody was just going for the same goal. And it was a magic moment, truly. So we arrived in Corona Tavern, and now we had a beautiful bouquet. We had applicable stuff, a Corona assay. We were trying to make it as user-friendly as we could. It was, yeah, arguably Corona was trending back then. And well, it was not so much fundamental. And so there we were with our little bouquet saying to industry, like, guys, we are still interested in getting together. And they just filled the floor with flowers. It's amazing what happened. So we were allowed to try new software features. They installed even new acquisition technologies in hopes of finding more biomarker peptides. We were provided with some of the latest consumables to optimize the sample preparation workflow, and they took care of measuring these samples in the state-of-the-art demo labs that they have. I mean, in my experience, we've never obtained any data as clean and good and sensitive as the one you get from industry. But more than anything else, I was surprised by how important they were for networking and basically credibility. If they say, like, check these guys out, they're doing a pretty good job, then people listen. So this is two features of the industry interaction I hadn't thought of up front. And I want to really have a special thanks to the Waters Innovation Response team, because of the immense amount of things they've done in order to help us. And so this is a very busy slide, but it's the same part as I showed earlier on the right. We managed to kind of straighten the curve by brute force. Try everything. Try different sample preparation protocols, try different instruments, try internal standards, the one from Polyquant here, try machine learning, try brute force. And it gave us a pretty good picture of where mass spec is standing if it comes down to this kind of assays. And we're very happy that very recently it also got published. You can see the author list is pretty big. So everybody interested in the actual development of the essay, feel free to go and have a look at the manuscript. I just highlighted Hans here, Hans Visters from Waters. Because of his position here, it's obvious the importance of him in this whole process and all the things I've mentioned earlier. So yeah, we couldn't have done it without industry. But as we were writing this up and trying to get it published and whatnot, I was very fortunate to be invited by the NHS, who in the UK was also really considering to make this essay clinically applicable, a MOSPEC essay. And they invited me to join this think process and just be there as an external reviewer. And this was led by Perdita Barron, but there's so many people there that are involved, and the efforts they pulled are considerably bigger than the ones we've done. And they managed to warp space-time and create a wormhole between the clinic and academia, because that's really how it feels. If you're in academia, you don't know how to reach out to the clinic, and they managed to do so. They got it all together. And if you want to read how they did it, the latest issue from The Analytical Scientist is describing that in pretty big detail. And on top of that, it contains art made from mass spectra. So that's awesome in its own right. That being said, they did it, and it's crazy. And what happened, I think what is a very important asset here was that a company called Cisco joined the effort. They had joined our consortium, and they joined NHS because they have this clever technology wherein they immunize rabbits with the actual target peptide you want to be using in the end of your assay. So they don't immunoprecipitate proteins. They capture the peptide that you want to be seeing on your mass spec in the end. And what happens if you do that is your curve, either way, your sensitivity goes up because there's less of an ion suppression here, but they can dig way deeper than anybody else because the background is gone. So you're ending up with almost a 0-1 kind of signal and basically landing near a CT of 30. So I think that calls for a celebration. And where we've been working with Cisco, they're amazing. And they too have helped us so much with reagents and trying to help them find the right targets and whatnot. So a great collaboration there too. Now the party really is getting crazy, since NHS found that they could have, well, they reached the sensitivity and a specificity of about 96% at a CT of 28. So really impressive effort. But there's a problem now, because they also looked at, they had the same samples analyzed by qPCR by two different labs. And you can see here in the x-axis is the average CT from these two labs. And on the y-axis, you can see the difference in CT value between these two labs. So you can already see it's a pretty big cloud. And I just want to point out, we are mass spec people, so MS2, we think it's MSMS. That's another name for their internal control. So it's not mass spec here. But you can see how big this cloud is. And if you think about it, 8 CT values, which is this line and everything above, that is 2 to the 8th, or a 256-fold difference in the estimated viral load. So if we say we're stuck at CD28, we need to also be able to ask, what do we need to benchmark against, and what are we benchmarking against? I'm not doubting the usefulness of qPCR here, but it's hard for us to get a good number on sensitivity and specificity if there's this amount of variability. So that's also why on June 24th with Waters, I'll have a short presentation on the fact that we amplify the signal and not the analyte. It, it's an idea that basically Perdita, for the first time, made me realize like it's important to detect what is in your sample before you've done anything to it, like amplifying. And we think that probably a mass spec will be more quantitative. We don't know that yet, but if anything, will never be as sensitive as qPCR. We cannot detect a single molecule, but then maybe we might be more quantitative up until a certain CT value. Okay, so summing up the Corona adventure, we ended up with the COVMS saying that if you have a saline buffer and you use a good internal standard like the one we got from Polyquant, again, a company with which we work together and who provided free consumables for us to make this possible. We then look at a few peptides, and at least the latest generation triple quadrupoles that we worked with, which was a Waters and a Sykes at that point, they were able to go down to roughly a CT25. Again, you'll need your heavy standard, the blue one here, to make sure that the red little bump is a bump, but it's feasible. And that's where we landed. Now, what happened at NHS, and we're still also trying to develop this assay in new ways, is, again, you need a standard. I just added Polyquant because that's the one we know. You don't need to use this one per se, but is to uncouple the assay from the transport media so that it can be applied to any medium that was used. Obviously, Cisco is now an indispensable part of the workflow. Basically, just 2 days ago, Waters launched their kit, which is entirely built on everything we've done before, what they've done before, obviously, in optimizing this assay as good as they can. And there's a very big issue with automation, as you might imagine. If you at one day want to be doing 20,000 samples, like PCR is capable of doing, we'll need automation, as they have. So there's a lot of interest in that area as well. But with yet another generation of instruments up and running, and with this kind of, well, clinically applicable by now, I think we can say clinically applicable workflow, we're going down to, say, detecting 50 attomoles of viral protein in a sample. So that is really, really close to CT30. And because they're so clean, we can probably squeeze the gradient down to 1 minute, having close to 1,500 patients a day on a single instrument. So that's the right side. Here is things that we are also working on, but that is way bigger. That is way more than what we've done. There is dozens and dozens and dozens of people working on this all together. And again, it's very nice to see it happen. But that brings me to the conclusion. This blind date with industry in Corona Tavern, was it just a fling or the start of a romance? I've put this blind date, the blindness of the date, on a scale kind of thing. And I mean, truth be told, we've been working with Waters for over a decade, so that's not really blind anymore. And going up in this way with Sykes, we've been working together for, what's that now, 7 or 8 years. And then Polyquant, we knew them up front as well. The most blind date we had was with Cisco, and it was a beautiful one. So Polyquant, you think now, how did you know Polyquant? Maybe some of you remember the YPIC challenge. This is a busy slide. It's just 3 snippings from an issue in UPA Open. The YPIC challenge number 2 was the one where Polyquant, free of charge, because they love the idea, expressed a protein that is a sentence in E. coli. And we sent that out to labs for others to try and decipher what was in the sentence. This was finally won by Lili Niu. Congratulations to her again from the Matthias Mann lab. And there's an issue in the YPIC Open Proteomics that describes all the details on this. So again, Polyquant was there, we knew them, and they came to the rescue once again. So is it a fling or the start of a beautiful romance? Well, at least with Polyquant and Cisco, we're looking into the idea of adding other viruses to the same assay. I mean, if you had that nasty swab in your nose, you might as well figure out what you actually have. For now, they just tell you it's not corona, but it could be something else. And it's not that big a deal, no disrespect, to have other viruses included in the same assay. It's perfectly feasible, and we're looking into this. So they keep on working together, and it's great. On the instrument side of things, let's just say I'll keep on plucking flowers. And I hear they have amazing stuff going on and a lot of new things heading our way. And we'll see. For now, it's complicated, and it's always going to be complicated. So I'm just going to share what I've learned from this. And it was quite a big journey. And sorry for bothering you with the entire journey, but I think first and foremost, just follow your passion for science. I mean, don't focus on the fact that you want to be working with industry. That is not the right focus. You want to be focusing on good science and then hope to meet industry somewhere along the way. But do realize that they can help you a lot. Again, this can go from huge discounts, enabling you to buy something you would otherwise never be able to, all the way down to whatever they could help you with. So I do see them as a valuable partner. But be patient and grow first. I myself was ambitious if you're young. I mean, 10 years ago, I wanted to become a hardware beta tester for the next MOSPEC. There was nothing, I had nothing to offer. That was not a realistic thing to want. I don't think it is already. So just be patient, I guess. And I'll be patient myself, obviously, also. And be interested in what industry is interested in. I mean, if you're working on a very specific biology of a very specific disease and using a ton of different techniques that you just want to spit out results, chances are that the MOSPEC family is not interested in teaming up, because you need to be interested in the actual technology, if this is something you like. But there's other things in MOSPEC where you can apply this to, I guess. I kind of learned to treat industry like I want to be treated myself. So I think really, at first they were these big capitalist organizations that would crush all the small academia people, that they don't turn out to be like that. So that is something I really started to appreciate. Make sure to give. Just don't sit waiting until they give you something. Make sure you have something to offer. This is a very personal one. If you come from a niche, it's harder to convince industry to invest in you. But on the other hand, there's fewer competition, or it's more manageable. So I don't know if that's a benefit or a disadvantage. I honestly don't know. But I do realize that for us, failing is a disappointment. And as young PhD students, we had or have a lot of those. But for industry, stakes are way higher than just disappointment. So again, it's something you need to realize if you're teaming up. And I think with that, there's so many people to thank. We already had a bit of a collaborative history, but what happened in the last year just exploded, as you might imagine. So by no means this list is exhaustive, and we're just immensely grateful for everybody, joining in the same quest. And I'm not saying, everybody has their own thing going, and it's just so great that we could touch base and then each go its own way again. And it was a very amazing period. So it was already advertised in the beginning, but make sure for the 21st of June event from YPIC to apply to your local proteomics society so that you become the one representing your country in a 5-minute pitch to show your work. And with that, this was my presentation. I'm open to any questions.
Okay, thank you very much, Maarten. This was a very nice talk. I would like to start with a shout out to the audience. If you want to ask anything, I forgot to mention this in the beginning, just put it into the chat or write me directly and I will read out your question. And I will start with the first question that actually came from the audience. And this was at the slide where you have shown all the problems that come with doing mass spec on histones. And someone in the audience asked if ion mobility would actually solve your problem. I think you gave a short answer to that already, but can you sum it up again?
No, you could, yeah, you could have gotten it from that slide saying unpublished. It turned out to be way harder than we hoped for. I literally spent 2 years diving into data and trying to figure out whether we could make it work. The short answer is we will be able to make it work, but it was way harder than we anticipated. So I would bet on the next generation ion mobility devices to really make a difference. And then you really need to know how that data structure looks like and how to handle this kind of data. So it's going to be a game changer, but it's really hard.
Okay, then there's another question from the audience. Regarding immunoenrichment with cis-kappa, do you target multiple peptides? And was cis-kappa preferred over an enrichment at the protein level like the MSIA approach?
Yeah, so generally speaking, cis-kappa and people working with it would argue that 2 to 3 peptides is the ideal way forward, and that's what most people are doing. We are still keeping an open mind, trying to look at more targets still. But protein IP has its own benefits. It has the disadvantage of being more unpredictable, capturing proteins as a whole. If there's conformational changes, if there's PTMs, if there's other stuff sticking to it, it's harder to make sure that your purification works. But the huge benefit of protein IP is that once you've captured the protein, you can digest the protein and start looking for mutations. You can start picking up variants of concern, something you cannot do with cis-kappa because you're only targeting like 2 or 3 peptides. And I mean, if the mutation is in that peptide, you won't capture it. And if it's not, yeah, you don't know. So both are very valuable.
Okay, cool. So now I have to, and I was asked this also from the audience. Also, one question I have to ask, how are you outperforming the PCR assets? How is your assay applicable?
Yes, that question always comes. Do we outperform qPCR? No, we don't at all. Is that a reason not to do it? No, not at all. So we are not as sensitive, as I clearly mentioned, but we are versatile. Mass spec as a technique is versatile. I'm speaking in the name of the field. The thing is, for example, picking up variants of concern is something we could add possibly relatively easy. Looking at other viruses, same there. We're still in a learning process. I mean, mRNA vaccines were made in the last year. That is 10 years of research was preceding that. So we are still in an early phase. But as we go, the development goes quicker and easier. And mass spec is just more versatile. You can even imagine having a mass spec sitting in a clinic, doing like 100 patients, and then moving to stuff coming in from hepatology, and then something coming in from another department, and you're just biomarker shooting everything around. And that is probably the future of mass spec in the clinic, the versatility.
Okay, cool. That's what I think was actually my next question that you answered now. There's another question from the audience. So considering the theme and your comment with respect to new generations of instruments, will timsTOF become a new lover for your research and maybe for industry as a whole, or is it too new to bet on it?
It's the metaphor of a relationship holds pretty good here. If I see another beautiful girl, am I interested? Yeah, but I'm in a relationship. So no, I think timsTOF Pro is an amazing instrument. Everything I've seen from it or of it is amazing. So objectively speaking, I can say the same thing about an Orbitrap. It's an amazing instrument. So no worries. iMobility is here to stay. So any instrument that has that is probably going to be a very interesting candidate. And yeah, again, my type is QTOF. It's a QTOF, so it's hard to resist. But for now, I can.
Okay, and if there are no more questions, I would like to finish with one. Your journey seemed like one that once it started, it just kept on rolling. And also I like this interaction with a game developer, a quite large one. Those are nerds and I can see how they like your challenge. But how do you start off connecting with industry? What did you do to make it going or get it going in the very beginning?
It's a very good one. If there is one, there is one memory at least that I have with Hans Visters. You would be surprised. There he is again, sitting at ASMS. I don't know what edition, 2006, 7, 8, somewhere. He telling us that Waters was working or had developed MSE, MS to the E, and for the first time, I was ripped out of the DDA convention and started seeing them think in other ways I never had thought about. That conversation went well. Like we felt like he saw the enthusiasm, and it took another 10 years before we really got this going. So I don't know if you need a specific point. I think people from industry need to see that you are really passionate about what you're doing, and if then you find somebody that believes in you, one day they just come back and say, how have you grown since? And that's how it happened to me, at least. So a big thanks to Hans from my side.
Okay, so I guess passion and, as always, networking. Okay. Yeah. Thank you for this brilliant talk, Maarten. I had a blast, and I'm sure the others had as well. Thank you for your time. And I wish all of you a pleasant day afterwards.
Goodbye.
Bye-bye.